Hello and namaste to all of you. This is Jayvin Shamasan from the Department of Physiotherapy and Diagnosis from Vidyapeeth University. In last lecture, I introduced you about the vitamins. So today we start with one of the most important vitamin called as vitamin A. So it was basically isolated in 1913, and it was basically identified in carotenes. So vitamin A is a fat soluble vitamin, and the active form is present only in animal tissues. Now the provitamin beta carotene is present in plant tissues, and beta carotene has two beta ionic rings, which is connected by polyprenoid chain. Now one molecule of beta carotene can theoretically give rise to two molecules of vitamin A, but it may produce only one in biological system. So all the compounds with vitamin A actively are referred to as retinoids, and they are polyisoprenoid compounds with beta iono cyclohexanide ring form. Now these three different compounds with vitamin A activity are retinol, that is vitamin A alcohol, retinol, that is vitamin A aldehyde, and retinoic acid, which is vitamin A acid. Now, the retinol, it may be basically reduced to retinol by retinol reductase, and this reaction is readily reversible. So, retinol is oxidized to retinoic acid, which cannot be converted back to other forms. Now, the side chain, it contains alternate double bonds, and hence many isomers are possible. So the all trans variety of retinol is also called as vitamin A1 and is most common. So vitamin A2 is found in fish oils, and has an extra double bond in them. So biologically important compound is 2,6-retinol. Now what about the absorption of vitamin A? Beta carotene is cleaved by dioxygenase to form retinol. Now the retinol is reduced to retinol by an NADH or NADPH dependent retinol reductase, which is present in intestinal mucosa, and intestine is the major site of absorption. The absorption is along with other fats and requires bile salts. So in biliary tract obstruction and steatorrhea, vitamin A absorption is reduced. So within mucosal cells, the retinol is re-esterified with fatty acids incorporated into chylomicrons and transported to liver. So in liver sterate cells, vitamin is stored as retinol palmitate. Now, how can it be transported from liver to tissues? So the vitamin A from liver is transported to peripheral tissues as trans-retinol by retinol binding protein, which is RBP. And the one molecule of RBP binds to one molecule of retinol in case of vitamin A deficiency, and the RBP levels in blood will fall. So now the question arises, how will it be uptaken by the tissues? So retinol or RB and RBP complex that was formed, it is specific to receptors on the retina, skin, gonads, and other tissues. So the RBP, it does not enter in cell and inside the cytoplasm of the cell. So vitamin binds to cellulose retinoic acid binding protein, and it finally to hormone responsive element of DNA. So the genes are activated. So one of the important, since vitamin A has many roles, but one of the most important role is vision. In vision, so there is walled visual cycle. So generation of nerve impulse. So Wald was awarded Nobel Prize in 1967 for identifying the role of vitamin A in vision. So Rhodopsin, which has molecular weight of 35,000 daltons, it plays a pivotal role in vision, and it is a membrane protein found in photoreceptor cells of the retina. So the rhodopsin is made up of protein opsin and 2-cis-retinol. And when light falls on the retina, the 2-cis-retinol, it will isomerize to all trans-retinol. and a single photon, it can excite the rod cells. So the photon produces immediate conformational change, and the unstable intermediate produced are rhodopsin to beta-rhodopsin to lumirhodopsin and meta-rhodopsin 1 to meta-rhodopsin 2 and finally to opsin and all trans retin. So each of these intermediates have a lifespan of few picoseconds or microseconds. So the all trans retinal is then released from the proteins. Now these visual segments are G protein coupled receptors, and 2-cis retinal, it locks the receptor protein and its inactive form. So the isomerization and photoexcitation will lead to activation of G protein and generation of cyclic GMP. Now this cyclic GMP acts as the gate for cation specific channels, and transducin is the G protein and retin. Now the nerve impulse thus generated in the retina is transmitted to visual centers in the brain, and the signal is transmitted by phosphorylation of a serine residue of activated rhodopsin by an enzyme rhodopsin kinase, so that the inhibitory protein beta-arrestin can bind and inactivate rhodopsin. So how will this 2-cis retinal be regenerated? So about the regeneration, we'll take in next class. Until then, if you have any doubts regarding this part, you can ask. And until then, let's take care.